Morning, we are ready to begin this graduate session in biotechnology of human reproduction, and I welcome and greet the two candidates, Teniola and Anna Chiara, who will be discussing their thesis remotely for the pandemic reasons, and I hope there are no technical problems. And so the first candidate is Teniola Adewale, and the supervisor is Prof. Collodel. The second supervisor is Prof. Moretti. The examiner is Prof. Piomboni. The title thesis is Characterization of Spermatozoa with Severely Reduced Motility by Transmission Electron Microscopy and Immunocytochemistry. And I ask you, Teniola, to read the first statement.
Okay. I, Teniola Adewale, matricular number 098725, enrolled in the Corso de Laurea, Laurea Magistrale in Biotechnologies of Human Reproduction, considering the decree number 461/2020, protocol number 52533 of the Rector of the University of Siena, dated March 11, 2020, which authorizes the online procedure for the final exam, graduation, and public proclamation. Being aware that, according to Article 76 of the DPR 445/2000, false declarations, falsity in public documents, and use of false documents are punished according to the Penal Code and to the current special laws on the subject. I, Adewale Teniola, own responsibility to accept to perform my final examination via online procedure, and that I will not take advantage of any external help during the performance of the examination. Thank you.
Please, Teniola, your presentation, okay?
Thank you. Good morning, everybody. My name is Teniola Adewale, and the topic of my thesis is characterization of spermatozoa with severely reduced motility by transmission electron microscopy and immunocytochemistry. So approximately 1 in 5 couples have fertility problems, and this burden of infertility is being shared by the couple. Sometimes it is caused by male factor, sometimes female factor, and in some cases it could be mixed factors. And in some cases as well, it could be due to unexplained factors. For my thesis, I focused on the male infertility, and the causes of this can be wide-ranging. An example of that is spermia, which is characterized by a severely reduced sperm motility. So the spermiogram, which is the first-line analysis that we carry out when we encounter patients with infertility, it does not give us the identification of the full range of alterations that may be causing this astenospermia. And the assisted reproductive techniques that we might want to imply, the outcomes can be impaired depending on the causes of this astenospermia. So investigations like transmission electron microscopy and also immunocytochemical studies enable us to overcome this limitation, and also to characterize sperm pathology into non-systemic defects and systemic defects. So the non-systemic defects, they are more common, and they cause different degrees of reduced sperm motility from mild to moderate or even severe. And there are a combination of defects that affect the sperm tail. So when we are talking about the non-specific flagella abnormalities, they could alter the number or even topography or organization of the axonal structures. And these defects can be generally associated to different disorders. For example, infections like epididymitis can lead to necrospermia, which is also an example of non-systematic defect. And even hormonal imbalance or variability can also be examples of disorders that can lead to this defect. For the systemic sperm defects, they are very rare, but they cause severely reduced motility. And it's important to know that they are of genetic origin and they have a predominant phenotype that is common to everybody that has this defect. So the defect may affect the head or even the head-tail junction or the flagellum of the spermatozoa. Examples of this can be dysplasia of fibrous sheath or even head-tail attachment defect, even proboscistemia, which is known as round-headed spermatozoa. So what is the purpose of this study? It was aimed at the characterization of spermatozoa with severely reduced motility in 3 infertile patients using the transmission electron microscopy, which I will be referring to as STEM moving forward, and immunocytochemistry. So we have our 3 patients, all young men of similar history of infertility, who have normal sexual development and medical history. They were not of consanguineous ancestry, and none of them were smokers nor alcoholics. Then we have our control as well. The semen of a 35-year-old man with proven fertility was used as the control height microscopy level, which was done 3 times at 3 month interval. So the analysis of the sperm concentration, the motility, the morphology, and the vitality were carried out. But it still did not allow us to understand the full range of morphological defects that may be affecting the sperm organelles. Another investigation that was carried out was the acridine orange assay, which highlights the integrity of sperm DNA by measuring the susceptibility to acid-induced denaturation. So in this picture, we can see some highlighted in green. Those are the sperm heads that contain the intact double-stranded DNA. And those highlighted in red are those that already contain the denatured or single-stranded DNA. So immunocytochemistry study was also carried out, which helped us to study in situ the biomolecules, sperm proteins, like in this case, we were interested in the tubulin protein, which is a protein that forms the microtubule, and it runs along the whole length of the sperm tail, and also the centrin-1, which is a very important protein in the connecting piece that helps in the head-tail attachment and also mitotic spindle assembly during fertilization and early embryo formation. So for our primary antibodies, we used anticentrin-1 and anti-beta tubulin to target the proteins that we want. And then a secondary antibody was also used to amplify the proteins that we want to localize. Then also transmission electron microscopy was done to allow the study of the ultrastructure of the spermatozoa, and also to characterize the structural sperm defects that may be present. So what are the results that we got? In the fertile control in the fertile control man, the semen parameters under spermiogram were seen to be more than 50, to be above the 50th centile using the WHO 2010 reference. And also the acridine orange analysis showed that 98% of this man's spermatozoa contained double-stranded DNA. Looking here, we can see the TEM picture, and this is how a normal spermatozoa is supposed to look under the transition electron microscopy. We can see the head of the spermatozoa, which is covered with the acrosome, and in the sperm head, we can see the compact nucleus, and also the tail that runs down. We can see the mid piece that is covered by the helix of mitochondria, and then the principal piece and going downwards. And the also, we can see here the immunocytochemical study, the localization of the tubulin. You can see it in green that runs along the whole length of the sperm tail. And then the localization of centrin-1, these are 2 spots at the base of the nucleus in the sperm head. So that is the normal localization of the centrin-1. So for our patient 1, the sperm concentration in this patient was about 15 to 18 million. But the interesting thing is that there is no progressive motility at all in this patient. Then going on to look at the vitality, only 10 to 20% of this patient's spermatozoa were viable. That means more than 80% of this patient's sperm are dead. So this can already bring us to the diagnosis necrospermia. But to further confirm this, we use transmission electron microscopy, and we can see the disrupted chromatin here, also reacted acrosome. Looking at picture B here, we can see the swollen mitochondria, and all of these are all processes of necrosis going on. Staining with immunofluorescence, we can see in red the tubulins. We can see a lot of isolated tails and also coiled tails present. Then for the century 1 localization, we can see the red spots, but their localization is mangled up together. And this may be as a result of protein degradation that is occurring due to the necrotic process. Also, acridine orange analysis in this patient only showed 20% of the man's whole spermatozoa containing double-stranded DNA. For our patient 2, the sperm concentration is about it falls under the 10th to 50th centile, but we can see that 85% of this patient's spermatozoa are acephalic. And looking at the table down here, only 7% of complete spermatozoa have rapid progress motility. The acridine orange result, although, shows that 95% of this man's observed sperm and even the isolated head showed double-stranded DNA. But looking at the TEM picture, we can see the head-tail angle, we can see the head-tail connection defect, and also the cytoplasm residue here, and a whole lot of isolated heads and tails. Going on to staining for tubulin, we can see a whole lot of isolated tails all round, and the century 1 in this patient was totally absent in the head of the sperm. So for our patient 3, the sperm concentration was about 20 to 28 million, but the progressive motility in this case as well was very low, almost totally absent. The interesting thing is that the vitality of this patient is about 80 to 84%. And then the morphology is just 1 to 2%, meaning that in this patient, there were a whole lot of abnormalities going on in the head region, in the tail region, and even in the head-tail junction, which brings us to the diagnosis: multiple morphological abnormalities of the sperm flagella. As we can see in this picture, a whole lot of isolated, you can see isolated tails, and even here we can see where the fibrous sheath runs along the whole length of the tail. And then staining immunofluorescence for the tubulin, we can see the angled head and tail, and also coiled tails are present, and a whole lot of abnormalities in the tail of this patient's sperm. So for the centrine, some of the sperm heads had multiple centrines localized at the base of the nucleus, although some of them still contained, the centrine 1, many of them the centrine 1 were localized properly. So in conclusion, the immunocytochemistry and transmission electron microscope enabled us to diagnose the causes of the astenosospermia that affected the 3 patients, because although they suffer from the same cause of infertility, which is the astenosospermia, the underlying causes of this reduced motility was different in each patient. As we can see, in patient 1 was necrosospermia, in patient 2 was acephalic spermatozoa, and in patient 3 was multiple morphological abnormalities of the flagella. So the result of this thesis focused also on the importance of implementing investigations like the TEM and also the immunocytochemistry to the study of the connecting piece, and also to the protein century 1, which is very important in the connecting piece. So this study can help us to identify defects in the sperm centriole and to characterize their impact on male infertility and embryo quality. So in summary, this is a representation of all that I have been talking about: the patient and the results, and the materials and methods and the results. Thank you very much. That would be all. Thank you for your kind attention.
Thank you, Teniola, for your presentation, and a correct diagnosis of male infertility is a relevant issue to be investigated. And I ask the Professor Piomboni if she wants to make some questions.
Yeah, very good, Teniola. Very, very interesting report, because you are analyzed. The analysis was done in 3 main categories that are, I think, the most frequent category of defect in infertile male, even if some of them can have a genetic origin, for example, detached tail or isolated head, as you prefer. But very interesting. To curiosity, you know if any other protein can be localized at the connecting piece together with centrine, for example, and if you know if it is not in the literature, if there's a kind of patients with such defect of sperm can fertilize by ICSI, for example. ICSI bypass all the problem of motility because you inject the sperm into the oocyte. So if you know that in the literature are reported cases about fertilization and embryo formation after injection of this kind of sperm.
Okay, the first question about proteins localized in the connecting piece. There are other proteins. For example, we have the centrosomal protein 131 that helps with the microtubule trafficking, and it also helps with the protein transfer into the tail. And as well, we have other proteins, SPATA6. This also helps in the development of the connecting piece itself. Then another protein is the cilia and flagella associated proteins, which mutations of these proteins can actually lead to the diagnosis of that patient 3, which is the multiple morphological abnormalities of the flagella. So all of these proteins are located in the connecting piece as well. Then
so the connecting piece is very rich of protein.
Yes.
There are many proteins.
Yes.
It's very, very important place in the sperm because of the contribution of the centriole to the embryo, for example. So it's a very, very important region also for ICSI, I think. Okay.
Yeah. So for the possibility of ICSI, because this connecting piece is very rich in protein, take, for example, the centriole one that I was talking about. It is important not only in the head-tail attachment, but also in mitotic spindle assembly.
Yeah.
During fertilization and early embryo development. So if this centriole one is not localized properly, or if it is absent in the head, go ahead to do ICSI. It could lead to maybe birth defect or even abnormal embryo development. So in this case of patient 1 and patient 2, it is not advisable to go ahead, in my own opinion. But in patient 3, it is possible to still find spermatozoa that has correct localization of these proteins, and we can go ahead to do ICSI. So the outcomes of ICSI in each patient is varying.
Yeah, the situation may be variable also among patients with the same or similar sperm defect. So case for case, the embryologist, I think that in the future can choose the right sperm to be injected after a correct diagnosis anyway. Okay, thank you, Teniola. Very, very good, very interesting.
Thank you very much.
Thank you, Teniola. The second candidate is Anna Chiara Crosato, and the supervisor is Prof. Luddi. The second supervisor are Nardi, Prof. Nardi and Prof. Fabro. The examiner is Prof. Collodel. The title thesis is Could Made Age Influence Semen Quality, and are code for analysis and comparison of morphokinetic parameters. And Anna Chiara, I ask you to read the first statement, please.
So, the undersigned Anna Chiara Crosato, matricola number 098491, enrolled in corso di laurea magistrale in biotechnologies of human reproduction, considering the decree number 461/2020, protocol number 52533, of the Rector of the University of Siena, dated March 11th, 2020, which authorizes the online procedure for the final exam, graduation, and public proclamation, being aware that, according to Article 76 of the DPR 445/2000, false declaration, falsity in public documents, and use of false documents are punished according to the Penal Code and to the current special laws on the subjects, declares under her own responsibility to accept to perform her final exam via online procedure, and that she will not take advantage of any external help during the performance of the exam. Klagenfurt, Austria.
Thank you. Please, your presentation.
Mhm. Okay. So, good morning to everyone. The main question of my thesis is, could male age influence semen quality? An air code for analysis and comparison of morphokinetic parameters. First of all, what is the infertility? The correct definition of infertility is the inability of a couple to conceive even after 1 year of unprotected sexual intercourse. And it affects at least 180 million of couples worldwide. The main factor is a contributory in about 30% of all cases of infertility. And although the effect of maternal age on fertility is quite clear, it is still unknown if paternal age also could affect fertility. So, the main aim of this study is to discuss effects of male aging on semen parameters, giving also an explained, ready-to-use R code to analyze clinical data. Furthermore, the available code could be used as a starting point to include more data, such as female parameters and outcome in art. The problems in male factor fertility may be due to changes in semen quality parameters, and the routine semen analysis is the initial step of male infertility evaluation, but it provides no insight into the functional potential of the spermatozoon to fertilize an ovum. So we have two different kind of main examinations. First of all, the microscopic examination, which include the examination of the semen appearance, liquefaction, volume, viscosity, and pH. Then we have also the microscopic investigation with the sperm concentration, with motility and morphology, and also with assessment of the seminal leukocytes and immature germ cells concentration. And we have different findings about the possible relations between the increased male age and general semen parameters. So first of all, we have studies which found no changes in the volume, concentration, motility, and morphology. On the other hand, other studies has found changes linked with increased male age. In particular, they found an increased time to pregnancy, a decreased pregnancy rates, and also a decrease in semen concentration, motility, and morphology parameters. Then another aspect is the computer software, which have become indispensable in the meta-analysis. In the last decennia, many programs have been developed. In particular, R and RStudio are free language and software for statistical computer and graphics programming. And in particular, R is the industry's leading analytical tool, commonly used in data modeling and statistics. Thanks to them, it's possible to manipulate and present information in various ways. And also, it also offers instruments to install all the package automatically, according to the user's needs. So, my study is differentiated in different steps. So first of all, the subjects. This study is a non-interventional retrospective analysis of about 50,000 consecutive semen analyses from January 2011 to September 2021 in TFP Kinderwunschklinik in Klagenfurt. Each patient produced a semen sample into sterile plastic containers, and samples were allowed to liquefy at 35 degrees for at least 10 minutes. Then follow with the semen analysis, so the microscopic evaluation of the appearance, liquefaction, viscosity, and pH values, and then the microscopic evaluation with the concentration, motility, and morphology. Then with the data preparation, so the set of the working directory of R, the upload of the file, with the removing of the null values, the reorganization of dataset, and the categorize of all the data. Then the data visualization, so the proper use of histograms, box plot, and bar charts according to the kind of data and the aim of the analysis. And at the end, the real data analysis, so the normality of the data, the multiple linear regression, and the final plot. So let's start with the first parameter. The first parameter is the male age. So in the first picture, it's possible to see the code of the entire coding, then the box plot, and then the histograms. And thanks to that, it's possible to obtain the mean of this value, so 38.4 years, with a range between 18 and 71 years, and with the majority of data between the 34 and 43 years. Then each parameters was compared with similar study, and this comparison is made for each parameter. Then we have the volume with a mean of 3.1 milliliter and a range between 0.1 and 16.8 milliliter. And we have also the concentration. In these graphs, the red lines indicates the inferior limit for a normal sperm count range. In particular, in this study, the 0.05% of all the samples were azoospermic, while 18.6% were oligozoospermic. Then we have the progressive and the total motility, which present similar values in percentage. And in the graph, it's possible to see the blue lines, which represent the 32% in the progressive motility histogram and the 40% in the total motility histograms, to underline the limit of the astenozoospermic samples. Then the morphology. In this case, the morphology is divided in good morphology with values are on the 4% and bad morphology under the 4%. In this case, we have the 34.2% percentage of the bad morphology and the 65.8% of good morphology, with a mean of the normal sperm morphology of about 8%. Then about the normality of data, it is calculated through the Sapphire test and the QQ plot. And we have found that the morphology and the concentration are not normally distributed, and so it's difficult to analyze. Then I found that the volume and the total motility are almost normally distributed, and the male age and the progressive motility are normally distributed. So, about the coding, this is the linear regression and the final code. Thanks to the coding, it was possible to calculate for each couple of parameters different values. So the couples were male age and the volume of semen sample, the concentration, the progressive and the total motility, and the morphology. So the code is a bit scary, but it's with this one, it was possible to calculate different values. The values are these ones. So the p-value and the confidence interval with the Pearson correlation, the intercept, the slope, the R-score, and the p-value of R-score. So maybe a graphic could help because, okay, these are all the values and it's better than the code, but it's still a bit difficult to understand the meaning of all these numbers. So these are all the graphics that is possible to obtain thanks to the values. And we have found that the best fitting lines in red, which are drawing basing on the intercept and the slope values. Then there is the confidence interval lines in orange, which depend on the uncertainty in the creation of the model. And then we have also the prediction interval lines in green, which depend on the uncertainty in the creation of the model and the uncertainty due to the differences among individuals. And you can say, okay, nice graph, but so what is the main aspect of these results? So we have, there is three main points. First of all, the p-values are significant, while the R-scores are very low. And this is due to the fact that data are very dispersive and not always normally distributed, and the correlation is low. Then there is the statistical relevance, but it's not possible to use this model to predict values of a selected parameter basing on male age. And at the end, these results are in line with other several studies, which also show a relation between the sperm parameters and age. So, in conclusion, this retrospective study suggests that the male age impairs semen parameters, and furthermore, it gives a complete, really usable R code to analyze data coming from every potential database. Further studies are required to investigate more consistently if male age has a relation with infertility and outcome in art. And then, a special thanks to the TFP team, and thank you all for your attention.
Thank you. Thank you, Anna Chiara, for your presentation. And I have only one clarification. And the last year, a paper about the influence, including the age, a paper on the influence of male age in semen parameters has been published. And in this paper, the authors considered some confounded value. And I want to know if you, in your work, have you considered smoking habits or varicocele or other, because you present a group of infertile men, but the age may be influenced also by different pathology associated with male infertility.
Yes, in fact, this aspect could be very important. And in fact, when we prepare the data for the analysis, so the first step before every kind of analysis, we have to differentiate the data and analyze them. So we have to understand the weight of the data. So which is the starting point. So we have to normalize everything and to understand which parameter could affect more or could not affect or could which could be more impact on the results and which not. And also, for example, if we want to improve the analysis with also the female parameters or with the art outcome, we have also to consider all these aspects.
Okay, thank you.
Can I say one word to underline that is a very, very new approach to the evaluation of so big amount of data in a male infertility, male sperm, male parameters, and so on. I think this is a very, very useful way to analyze the data to obtain significant results. And I would like to thank also the structure that hosts Anna Chiara. And I hope that Anna Chiara will have a very, very brilliant future in
Thank
this
you.
field. Many compliments. Thank you.
We leave this room to make our own assessment evaluation. Thank you. We are ready for the proclamation. Okay. Dr. Teniola Adewale,
Yes.
the committee, considering the results of the academic career, has approved your degree exam with a mark of 106 out of 110. Therefore, on behalf of the Rector Magnificus, we proclaim you, in the name of the law, Master Doctor in Biotechnology of Human Reproduction. Congratulations, Teniola. I ask you,
Many congratulations.
I ask you
Thank
to
you.
read the second assessment.
Okay. I, Teniola Adewale, matricular number 098725, enrolled in the Corso di Laurea Magistrale in Biotechnologies of Human Reproduction, considering the decree number 461/2020, protocol number 52533 of the Rector of the University of Siena, dated March 11th, 2020, which authorizes the online procedure for the final exam, graduation, and public proclamation, being aware that, according to Article 76 of the DPR 445/2000, false declarations, falsity in public documents, and use of false documents are punished according to the Penal Code and to the current special laws on the subject. I, Adewale Teniola, own my responsibility that I participated in the graduation exam on the 16th of December 2021 during the final exam session of the academic year 2020-2021 for the achievement of the final qualification, that I accept the evaluation and the final mark with which I am declared Doctor Magistrate.
Thank you, Teniola. Congratulations.
Thank you.
Dr. Anna Chiara Rosato, the committee, considering the results of the academic career, has approved your degree exam with a mark of 110 cum laude out of 110. Therefore, on behalf of the Rector Magnificus, we proclaim you, in the name of the law, Master Doctor in Biotechnology of Human Reproduction. Congratulations.
Bravissima, brava Anna Chiara.
Anna Chiara, I ask you to read the second assessment, please.
The undersigned Anna Chiara Crosato, matricular number 098491, enrolled in the course of laurea magistrale in Biotechnologies of Human Reproduction, considering the decree number 461/2020, protocol number 52533 of the Rector of the University of Siena, dated March 11, 2020, which authorizes the online procedure for the final exam, graduation, and public proclamation, being aware that, according to Article 76 of the DPR 445/2000, false declaration, falsity in public documents, and the use of false documents are punished according to the Penal Code and to the current special laws on the subjects, declares under my own responsibility that I have participated in the graduation exam on 16 December 2021 during the winter exam session of the academic year 2020-2021 for the achievement of the final qualification, and that I accept the evaluation and the final mark with which I declare the Teresa Magistrale in Biotechnology of Human Reproduction.
Thank you, and we wish you to fulfill your expectation now.
Grazie.
Thank you, bye bye.
Thank
Grazie a
you.
tutti.
Ciao, grazie a tutti,
Ciao.
grazie.
Grazie.
Ciao, complimenti
ciao.
di nuovo. Anna Chiara, poi ci sentiamo, magari. Ciao Segnola.
Ciao.
Ciao. Bene, io mi disconnetto e semmai mi riconnetto tramite il telefono, perché ora devo scendere a corsa.